Hello students, so last class we have already started with a new chapter. What was the chapter? Biotechnology principle and processes. So this is the continuation of the same, an interesting chapter. See to it that your people are understanding it in a proper way. Okay, very interesting, at the same time bit difficult to understand. Anyhow, let's go on, carry on. Okay, so last time it was an introduction I gave about our DNA technology. So the tools for recombinant DNA technology, we listed out 5 tools. Out of that, one of the tool was what? Restriction enzyme, isn't it? DNA polymerase, ligases, vector, host, other ones. Now we are going to discuss about what? Restriction enzyme in detail. Along with that, we will be also discussing one more topic, that is agarose gel electrophoresis. So these are the 2 topics that we are going to deal today, right? So what is a restriction enzyme? Listen students, a restriction enzyme is an enzyme which is usually used for cutting the DNA. What for it is used for? It is used for cutting the DNA. Other than that, they are having a different names. But the major aim in case of our DNA technology is that they are helping in cutting the DNA into different segments. So for cutting, since it is used, we call them as what? Molecular scissors. So last class we have discussed what is a restriction enzyme. Introduction was given. So this is a foreign DNA I am having. Imagine this is a foreign DNA I am having. Now in this DNA, I want a gene which is pest resistant. So here I can find out the gene which is pest resistant. So what I will be doing? I will be adding restriction enzyme. So this restriction enzyme will cut the DNA at specific places. Not just like that, they will cut. At specific location, the restriction enzyme will make a cut. And into that when they are making a cut, that DNA can be isolated, isn't it? How we will be discussing. So for cutting the DNA into segments, who is playing a major role? The restriction enzyme. Like this, I will get the gene of interest. Now the same, directly I cannot put it in the host, isn't it? So I have to first introduce where? Into the vector, make many copies, then only I will be going for the next step. So in order to introduce into the host, so before introducing into the vector, what we are doing? We are taking the plasmid, isn't it? Plasmid is an extra chromosomal DNA. There also I am adding what? The restriction enzyme. This restriction enzyme will make a cut in that plasmid, and this gene of interest is introduced where? Into this plasmid. Thereafter it is joined, right? So for cutting the DNA into segments, for cutting any part of DNA, these restriction enzymes are playing a major role. That's why the name is given as molecular scissors. What do we give the otherwise we call it as? We call the restriction enzyme as molecular scissors, right? So restriction enzymes are enzymes which cut DNA at specific sites into fragments, right? Yes, they belong to a class of enzyme called as nucleases. This is a one word question. So under which category does restriction enzymes category? We have transferases and all we have learned in the last isomerase, isn't it? So like that, under which category is this restriction enzyme following? It is falling under a category called as what? Nucleases. It is a one word question. Okay. Now, how this restriction enzyme was discovered? Actually, the name itself is telling restriction. So it is having some restriction. What is the restriction? What this restriction enzyme is having? See, in 1963, 2 enzymes responsible for restricting the growth of bacteriophage was discovered. 2 enzymes responsible for resisting means restricting the growth of bacteriophage. What is bacteriophage now? Bacteriophage is a virus. So imagine this is a bacteria and the virus is going to attack this bacteria. So the bacteria, what they are doing is that in order to protect themselves from this bacteriophage, what they are doing is that they are producing 2 enzymes. How many enzymes they are producing? 2 enzymes, right? One enzyme is helping in cutting the DNA into fragments, right? And another enzyme, what it is doing is that it is joining the methyl group. So they are helping in joining the methyl group to the DNA. Thereby they are helping in restricting the action of restriction enzyme. So restriction enzyme, the name, where from it is coming? It is coming from the discovery, this discovery. So what is the discovery? The restriction enzyme was isolated in 1963. How it was isolated? The bacteria is producing some enzymes. So what is this bacteria doing? This bacteria is producing some enzyme. How many enzymes? 2 enzymes. One is adding methyl group and another one is adding what? Cutting the DNA into fragments. Okay. So these 2 enzymes thereby are helping in preventing the bacteriophage to infect this bacteria. So bacteria are protecting themselves from the virus, the bacteriophage, by producing these 2 enzymes. Okay. So one is what? Enzyme is adding methyl group to the DNA, and other one named as what? Restriction endoreusine is cutting the DNA. By doing these 2 things, the bacteria is preventing themselves from the attack of bacteriophage, that is the virus. That's why the name is given as what? Restriction enzyme. It's not that they are cutting just like that the name is given. They are restricting the growth of bacteriophage on the bacteria. That's why the name is given as what? Restriction enzyme. Okay. Now when you are seeing how many restriction enzymes were discovered, now also you can find many are being discovered, right? So more than 900 restriction enzyme has been isolated from over 230 strains of bacteria. So strains, you know, different types. So around 230 strains. From there, how much amount of restriction enzymes are produced? Many different types of restriction enzymes are produced, and each restriction enzyme is having a particular site where they go and cut. Okay, that's why there are many different varieties of what? Restriction enzymes, right? Now this restriction enzyme, when we are giving a name, I have to follow some rules. So what are the rules? That's what we are going to discuss next. Okay. The first letter will be indicating the genus. So in this, the first letter is E. Example for this naming of restriction enzyme, we are going to take EcoRI is the one which we are going, EcoR1 is the one which we are going to discuss. Okay. So this is a restriction enzyme. EcoR1 is a restriction enzyme. The first letter indicates the genus. E is what? It is genus. What do you mean by genus? You know, when you are writing Mangifera indica, mango, Mangifera is the genus, indica is the species. Like that, here E is the genus and it is Escherichia. Okay, Escherichia, and CO is the species short form. CO is what? Coli. So Escherichia coli. Okay. And R is the strain. Different varieties of Escherichia coli are there. In that, one of the varieties R, representing what? The symbol R. Okay. So EcoRI. E is genus, Co is species name, R is the name of the strain, and 1. What is this 1? It is the order in which the enzyme were discovered. 2 means it is discovered next. 3 means it is discovered next. And from E. coli, I am getting different restriction enzyme. So number 1 is denoting the first enzyme which has been isolated. If it is 2 written, means what? This is a second restriction enzyme isolated from the E. coli, right? So this is regarding restriction enzyme naming. First name is the genus name. Second name is what? It is the species name. Third name is the third letter is the strain, and the Roman numeral, what is it is representing? It is the order in which the strain was discovered. Okay, so that is how the restriction enzyme is named. Now this restriction enzyme are of 2 types. What are the 2 types? One is exonucleases and another one is endonucleases. What is the difference between exonuclease and endonuclease? Imagine this is a DNA. So one is exo and another one is endo. Okay. So first one we are going to discuss about exonuclease. So if this is a DNA and if the cut, when I am adding a restriction enzyme, if the cut is made on the ends means you can find at the last base pairs on the tips. Okay, if a cut is made there, this is the cut, or here there is a cut, which means it is exonuclease, out towards the periphery the cut is being done, then we call it as what? Exonuclease. And what is endonuclease? Endonuclease means if the cut is made, so this is the DNA and the cut is made not on the periphery sides, it is made in between, inside. Okay, so in this the cut is made, this fragment is being cut. In that case I call it as what? Endonuclease. It is within the they are cutting inside. Okay, so that is the two different classes. One is exonuclease and another one is endonuclease. So exonuclease, they remove nucleotides. Nucleotides, you know what is nucleotide? Adenine, guanine, thymine, cytosine, you have learned. With sugar and phosphate, we call it as what? The nucleotides. So these nucleotides, when you are seeing, are cut on the periphery. Sequences are cut on the periphery, it is exonuclease. Okay, and from the ends. And if it is endonuclease, at specific location within the DNA. So this is the DNA in between. If the cut is made, then we call it as what? The endonuclease. Okay, specific regions are there inside the DNA where the cut is made. So like for example, you can find here, listen students, so just look into this diagram clearly. So in this diagram you can find the enzyme. So this is the enzyme cuts both the DNA strands at the same site, like vector DNA. You know what is vector? Vector, I told you, plasmid. Plasmid DNA is cut with the help of restriction enzyme. The same, Escherichia coli also cuts between the base pair only when the sequence is what? Present in. So like foreign DNA, this is the foreign DNA which I am going to take. So this diagram we have already seen. So one, I am going to take out what? The DNA, foreign DNA from any cell which is having that gene of interest. Okay, and another one is from the vector, I am going to take it out, right? So two restriction means, one, I am taking the foreign DNA, for that I have to make a cut in that cell. Along with that, in the vector also I have to take out that plasmid, and there also I have to make a cut. So two places I am going to cut. For that I have to use the same restriction enzyme, because the one restriction enzyme what I am going to cut, it will cut at the specific sequence, right? So whatever restriction enzyme I am using in this cell, the same restriction enzyme I also have to use in this cell. If I am using a different restriction enzyme, it will cut at different places. So it should not be the case. I have to use the same restriction enzyme. So that is again a very important role. Okay, so the restriction enzyme what we are going to use, it should be what? It should be of the same type. For the vector as well as the foreign DNA, the restriction enzyme what I am going to use is the same. Okay, now when I am adding the restriction enzyme into the vector DNA as well as in the foreign DNA, they are going to make a cut, right? So the cut will be made at specific places. You can find here also GAATTC, CTTAAG, that is the sequence here. Here also you see foreign DNA, also you see GAATTC, CTTAAG. Here also the same, GAATTC, CTTAAG. So this restriction enzyme will go cut here, and this same restriction enzyme will make a cut over here. Now when they are cutting directly in the center, they will not go and cut. Somewhat on the sides only the restriction enzyme will make a cut. After cutting, you can find there they are getting exposed, so that region is cut. Okay, so AATTC is cut, it is lost. Now here what is lost? The bottom one, CTTAA is lost, right? Now they are creating what? Sticky ends. Sticking end means that segments are getting exposed. There they have made a cut. The restriction enzyme has made a cut. Now what we will do, the next part is that now this foreign DNA, I told you when we are drawing what we have done. We have cut this foreign DNA we have taken. Now we have introduced into this plasmid, isn't it? The foreign DNA is incorporated into the plasmid. So for that what is to be done is that these sticky ends which are created, now they will join with one another and they will be pasted. They will combine together. Gene of interest is present here, that is going and joining with the plasmid. Okay, so for that this restriction enzyme is playing a major role. For joining this one, another enzyme is responsible. We have already discussed what was that enzyme? Ligase. DNA ligase is playing a major role, isn't it? So the ligase is playing a major role in joining the sticky ends together, right? So I will repeat again, restriction enzyme, hope it's clear. So the foreign DNA I have to take it out, isn't it? The foreign DNA, gene of interest. So in order to make a cut on the foreign DNA, what I will be doing? I will be adding one restriction enzyme. What I am going to add? A restriction enzyme. The same restriction enzyme where I will add it in the vector. Now in the vector also one cut is made, as well as in the foreign DNA also one segment is cut. These two segments, since the same restriction enzyme is used, they will make a cut in a similar way, right? So in the same place they are going to make a cut. Based on that, what will be created? Sticky ends will be created, one sticking on the other. So sticky ends will be created, and these two ends will combine together. For this combination, who is playing a major role? DNA ligase. Now this one, what I am getting is a recombinant DNA, rDNA. What do we call them? rDNA, because it is a combination of foreign DNA along with the vector DNA. That's why it's not a natural one which is already present. I am making it. So that's why this DNA is a recombinant DNA. Understood? So that is the role of what? Restriction enzyme, helping in cutting, and thereafter sticky ends are created, and with the help of DNA ligase enzyme they are being joined and rDNA is produced. What is rDNA? Recombinant DNA is produced, right? So restriction enzymes, when you are saying the restriction endonuclease, I told you endonuclease will be cutting in between, isn't it? So within a DNA strands they are going to make a cut. So when they are going to make a cut, there are specific regions where they can cut. That regions we call it as recognition sequence. Okay, so this recognition sequence are having a particular characteristics. Now what is the characteristics of this recognition sequence? They go and cut, recognize a specific region where they are going to cut. That region is called as palindromic nucleotide sequence. What is that region called? Palindromic nucleotide sequence. Okay, so what do you mean by this palindromic? Palindrome. Okay, so like for example Malayalam, if you are seeing Malayalam, when you are writing MALAYAM, isn't it? So if you are reading from the front to the back, you can find it is written as what? MALAYALAM. Now come from the back to the front, the same sequence it is written. So that is also MALAYALAM, which means if you are reading it from the front to the back, or from that side to this side, both the sides it's the same. Okay, so that sort of sequences we call it as what? Palindromic nucleotide sequences. So the restriction endonuclease, they will recognize these sort of sequences, the sequences where when you find from 3' to 5' end, as well as from 5' to 3' ends, the nucleotide sequences are having palindromic conditions. Okay, so like for example, you see here the example of a palindromic sequence is given. So what is that? First is 5'. Okay, so 5' GAATTC, 3'. Understood? So GAATTC. So at the same, so this DNA strand, one is 5' to 3', and another one is what? It is 3' to 5', isn't it? So when you are going for this 5' to 3', the sequence is how? GAATTC 3'. Now from back also you can find here also GAATTC 3'. Understood? So this sequences where the DNA is having the sequences like that, this side as well as this side, they are just similar. That sequences we call it as what? Palindromic sequences. Understood? So here example for palindromic sequence is given. So this is one DNA strand, this strand 5' to 3' is one DNA strand, 3' to 5' is another DNA strand, and they are having a sequence palindromic in nature, which means this is from front to back. You see GAATC. The from at the bottom, when you are seeing from 3' to 5', the thing is a bit different. It's from back to front, the sequences are one and the same. So this restriction enzyme will choose these sequences. There they are going to go and make a cut. Okay, so in that regions only they will be going and making a cut at the regions called as what? Palindromic sequence region, right? So restriction enzyme cut a strand a little away from the center of palindrome side. So they will just not go and cut in the center, a bit away from the palindromic sequence center. So here you can find where they are going and cutting. G is left, thereafter it is going and cutting in the vector DNA, isn't it? So they went and cut just after G they have cut. So what are the segments which were cut? AATTC is gone, right? So the same, the foreign DNA when you are seeing, they are going and cutting where? Just after G, right? So there also when they are cutting, you can find that segment is lost, right? So now what is created? Sticky ends are created. Same, AATTC on the top it is gone for vector, AATTC on the bottom it is gone for the foreign DNA. So what is created? You can find, since they are going and cutting at the similar sequence, these two regions are exposed. So here they are creating sticky ends. So if you go and stick something over there, they will get sticked too. Okay. So this one, this is the vector DNA is sticking with the foreign DNA at the sticky ends created by whom? EcoR1 is the restriction enzyme which is creating this one, right? Now what is the role of another enzyme? I told you, DNA ligase. They will join these two sticky ends, right? So when they are joining the sticky end, I am getting what? The recombinant DNA. Understood? So the recognition site is the palindromic sequence. That is what we have to remember. And give an example means you have to give the example as what? 5-GAATT C3-, 3-CTTAAG 5-. So what is G? You know, guanine. A is adenine, T is thymine, and C is cytosine, right? So these are the nucleotide sequences which we have already learned in class 11, right? Now moving on to the continuation of the same. Sticky ends form what? Hydrogen bonds with their complementary counterparts. These stickiness which is created is facilitating what? Action of the DNA ligase for joining both things. When they cut, the same restriction enzyme is used for cutting. The DNA fragments have the same kind of what? Sticky ends. So whatever restriction enzyme I am using for cutting the vector DNA, the same restriction enzyme we have to use for cutting what? The foreign DNA as well, right? Now one more thing what you have to know about the same is the restriction enzyme which was first discovered was, the first restriction enzyme that was isolated was HIND2. What is the first restriction enzyme? It is HIND2, and it is cutting at a specific region, that's a recognized sequence. That recognition sequence of HIND2 is just given here. So what is 5-? You know, G is what? Guanine, T is what? Thymine, PY represent to what? Pyrimidine. It can be pyrimidine, can be of two types. You know what are they? It can be either thymine or it can be cytosine, right? And another one is what? PU is representing what? Purine. So purine can be of two types. What are they? It is adenine and guanine, right? And A is what? Adenine, and C is cytosine. Okay. So these just one word, recognition site for HIND2. You have to remember, this is the recognition site for what? The HIND2. There only they will be going and making a cut. Which enzyme? HIND2 enzyme, right? So with this, restriction enzyme is getting over. Next we will be moving on to agarose gel electrophoresis. The next topic what we are going to discuss today is agarose gel electrophoresis. Okay. So what is the role of this process? Agarose gel electrophoresis, what is the role? Okay. So we will be discussing it. So this is a cell, isn't it? We have learned already in this diagram. This is a cell. Inside there is a nuclear membrane. Inside the nuclear membrane there is a DNA. So when I am adding this restriction enzyme, the DNA is cut into fragments. So out of these fragments, I want to take out only the gene, the segment which is coding for disease resistant, the gene which is coding for disease resistant. So the gene which is coding for disease resistant, only to take that segment out, I am using a process called as what? Electrophoresis. So that process we call it as what? We call them as electrophoresis, right? So the topic is separation and isolation of DNA fragment, that is DNA of interest or gene of interest. Okay. So this is done with the help of a machine called as electrophoresis machine. This is a machine. Okay, you can see it. Diagrammatically we will be seeing. So this is how the machine looks like. Okay. So when you are looking into the diagrammatic representation, it will be like this. So this is having a rectangular box, isn't it? So this one, inside this, actually what they are filling it is that, in this place what they are filling it is that they are taking agar-agar. Agar-agar, you know, it is a form of algae. So agar-agar with some amount of water they are mixing, making it as a paste, and then they are spreading it over here. Okay. So after spreading it over here, you can find there are blue wells like structure, means depressions are there. Okay, so you can find this is agar, the ash color is the agar spread out, and here 1, 2, 3, 4 you can find the rectangles, they are the depressions. Well like means inside the agar gel, what we are doing, we are making a depression. Okay, so what is done here, the DNA is taken. Okay, the DNA restriction enzyme is added. Now many DNA fragments are there. So the DNA fragments are collected and some fragments are put into first well, some fragments are put into the second well, some fragments are put into the third well, some fragments are put into the fourth well. So they are filling the DNA fragments in 1, 2 and 3 and 4 well, right. After this, what they are doing is that they are connecting it to electricity. Okay, so this well is connected to what, the electricity they are getting connected to. Okay, so when this setup is connected to electricity, what is happening? Listen students, this DNA fragments are negatively charged. What is the charge there? Negatively charged. So these negatively charged DNA will be moving when it is connected to electricity. The fragments, when you are seeing all the fragments which are present in one well, second well, third well, fourth well, on passing with electricity, the negatively charged DNA fragments will start moving where? Moving on the agarose gel as towards the positive electrode, they will start to move. Okay, so when they are moving, how they will be moving? They will be moving based on their molecular weight. Okay, so the smaller the fragments, the weight will be very, very less. So the smaller fragments will move faster and they will move away, isn't it? The bigger fragments which are somewhat having much molecular weight, they will be nearer to that of the well itself, right. So like that, the DNA which was with the help of restriction enzyme, many cuts were made. All these DNA, what is happening is that they are spreading on the agarose gel. So each and every one is what? The DNA fragments. Each and every DNA fragment, some fragments are coding for some characters, right. So like that you are getting bands of DNA. You can find all the fragments are separated based on their size. The smaller, the more they will move farther, and the bigger ones will be nearer to that of the well. Like that, the DNA fragments are separated. Now, how to see this DNA fragments? You cannot see it just like that. So, we have to stain it. So, agarose gel, what they used to do is that they used to stain this gel after passing through electricity. They used to stain it with the help of ethidium bromide. Okay, so they are using a stain, ethidium bromide. After applying, they are viewing it under UV radiation. Okay, so the UV light is given. So when UV light is passed, the DNA fragments will take up the stain, ethidium bromide, and in the presence of the UV light, they will be looking like orange bead like structures. Okay, so like that, orange color. So here it is given red anyhow. So orange colors will be seen. So we can find out that where orange color is there you can find the DNA fragments are there. Okay. Now the scientist will learn the DNA fragments. What is the DNA fragment? What are the nucleotide sequences? And from this, the scientists will find out. They already know pest resistant variety. What is the gene which is coding for pest resistant variety? So the nature of the nucleotide sequence in the gene they know. Okay. So like that, what they will do? Like for example, in all these, well, maybe this one. Okay, which one? This one is the gene which is coding for what? It is coding for pest resistant because they are having that nucleotide A, G, C, T distribution is like that. So that is the sequence. If it is identified, what they will do, they will take out this DNA only. Okay, only this DNA fragment. Which DNA fragment? This DNA fragment, they will be taking it out. So from the agarose gel, the process by which they take it out, after dissolving the gel, only that part they will be taking it out. That process we call it as elution. What do we call them? We call it as elution. That DNA fragment which is coding for pest resistant is now taken out. So in this way, what is done? We are separating or isolating the gene of interest. I repeat once more. Agarose gel is this setup Here agar is used as a medium, okay, just for transporting they are used as a medium. So what they are putting this what, agar is spread over here, okay. After that you can find what they are doing, they are cutting the DNA where the disease best resistant gene is present, that DNA, the foreign DNA is taken. With the restriction enzyme, many fragments are created. All these DNA fragments are poured where, into this wells. You can find blue into the wells, the DNA fragments are poured. Thereafter what they are doing, they are connecting it to electricity. Now DNA, since it is negatively charged, they will move towards what, the positive charge. So the smaller the DNA fragments, the faster they will be moving, okay. So the smaller ones will be moving farther and the bigger ones will be at the nearer to the well. After some time these DNA fragments will be separated and you can see it how you stain it with ethylium bromide, then view it under UV light. It will be like orange beads like structures. The bead like structures, genetic makeup is studied by the scientists and they will only separate out which one, the means the fragment which is coding for pest resistant is taken out. That process we call it as what? We call it as elution. So agarose gel electrophoresis, this is used for what? For taking out the gene of interest. We will just read it out. Separation and isolation of DNA fragments. The cutting of DNA by restriction endonuclease, we are cutting what? The DNAs are cut, okay, with the restriction enzyme. Now we are getting fragments of DNA. The fragment of DNA are separated by what? Gel electrophoresis. The DNA fragments are separated according to their size. The separated DNA fragments, how we can visualize? By staining with ethylium bromide, okay, and exposing to UV radiation. The DNA fragments, how they appear? They appear like orange colored bands, right? Now the separated bands of DNA are cut from the agarose gel. Yeah, the one which is gene of interest is taken out. That process we call it as what? Elution. These DNA fragments are purified and this is used in constructing what? Our DNA, okay. So for recombinant DNA making, along with the foreign DNA, the vector DNA are joined and we are making what? The recombinant DNA. So this is for today's class. Hope it's clear. Today we discussed two topics. One is the restriction enzyme and the second one is agarose gel electrophoresis. Both are very important. Hope it's clear. Thank you.